Hi. Good morning. See, there were people there who said hi, and there's people who said good morning. Because in health optimization, medicine, and practice, as you heard yesterday, hi is always a four-letter word. Today, we're talking about the Enteric Microbiota-Gut-Brain axis in the context of health optimization, medicine, and practice. Now, I'd like to disclose that I'm the founder of both of these organizations, Health Optimization Medicine and Practice, which is a nonprofit. We teach physicians health optimization medicine and non-physician healthcare practitioners health optimization practice on how to do health optimization. And the products of Prescriptions LLC, which is now held, a company held by Smarter Not Harder, is actually to help clinicians deal with more acute symptoms during patient or client care. For example, anxiousness and sleeplessness and pain. And we are essentially the main donors to Home Hope. So that's a disclosure for me. The general schema of everything that we talk about is this, is an enteric microbiota with a bidirectional communication with the gut, in a bidirectional communication with the organ. So it's enteric microbiota gut-organ communication. I actually created this slide because I was made to lecture on enteric microbiota gut-lung axis during the beginning of COVID. And as you can see here, the common theme is that all of these organs actually have malt. And I don't mean whiskey, because in health optimization, we have better drugs than alcohol. So MALT stands for what? Mucosa-associated lymphoid tissue, right? So you could see here, whereas before the other systems which we thought were sterile, like the ureters, etc., and placenta, they actually have microbiota now, as we have found out. So after this lecture, you will be able to discuss knowledgeably about the fundamental structure, functions, and new perspectives of the EMGB axis, enumerate the bidirectional communication pathways of the EMGB axis, mentally form the framework of Home Hope and its fundamental role in caring for the EMGB axis as a practitioner, be familiar with the technique of detection and correction of imbalance of Home Hope versus diagnosis and treatment of disease of illness medicine as applied to the EMGB axis, discuss knowledgeably about the enteric microbiota-brain interactions, neuroinflammation, and identify what's missing in today's research on the EMGB axis. And be up to date with the latest developments in the EMGB axis. Now, let's take the first part of this. This is structure, functions, and new perspectives. You guys already can always Google the structure and function, right? What I'd like you to remember from this section are really the perspectives that you should carry when you are in the clinic. Let's take a look at a quick structure. Many of you have more expertise than I have in this area. This is the distribution of microbial communities by weight. As you can see, 78% is in the GI tract. And here's the distribution on the right side of the slide, where you could see where the colon has 10 to the 11, jejunum 10 squared, and ileum 10 to the 7, 10 to the 8. So you could see where the distributions are of the gut microbiota. Now, the important thing here is to visualize it in several layers. The gut microbiota actually pre-process all of your food. Now, that's the key term here, is it pre-processes everything that you swallow. They try to pre-process even non-edible things that you try to swallow. So here, after they pre-process it and it passes to the liver, that's why when you take an oral drug, the liver tries to either activate or detoxify it, however the drug is configured. And then those raw materials are now then fed to the brain, to the heart, to the adipose tissue, the muscle, kidneys, and so on. All right, so the new perspective is the enteric microbiota is a postnatal organ. Okay, it's an organ that actually appears and grows after birth, right? It's seeded usually in spontaneous vaginal delivery through the vaginal canal. And as early as 2006, there were already some publications that this is the forgotten organ. Now, don't forget, microbiota is not foreign to you, right? It is part of what grows in you as an organ, except that it only grows after you're born. So here's a more recent, this is 2019 article, a representation of the gut microbiota in terms of prenatal, infancy, childhood, adolescence, adulthood, and aging. And you could see here on the lower part what they're actually doing, right, in helping to contribute doing. Now, Of course, the second part, that's your enteric microbiota. Remember in old textbooks, we just say gut-brain axis. That's already old, right? You have to specify enteric microbiota, gut-brain axis, right? In order to be more specific that the microbiota itself is an organ in the body. Now, the gut is actually a brain in itself, right? It has the lumen, of course, like the ventricles in the brain, enteric plexus, which is the main nervous system of the brain, which we call actually the enteric nervous system, right? And then there's submucosal plexus, and then the mesenteric. This is an old book that actually popularized the idea that it's the second brain. For many people, in fact, it is the first brain because evolutionarily, the brain is actually a later development in evolution. You had the gut first. So the functions would be nutrient absorption and immune defense, right? And you could see here underneath the intestinal lining, there's the Peyer's patches, right? And there's the M cell or the microfold cell that precedes it. And of course, there's a lymphoid tissue underneath. And this is called the induction patch, right? This is where most of the immune system actually interacts with anything that's in the gut lumen. Now, the new perspective that I'd like you to take home with you is that it's the largest immune organ. When you were in med school, it was the bone marrow, right? But we have, in the last 15 years, we now know that it's the largest immune organ, right? And you could see here the distribution, and with apologies for those who love Ayn Rand, who is GALT, gut-associated lymphoid tissue, that makes up 70% of the immune system. Now, let's go to the brain, the third part of the axis. The brain, we should always take a look that we have two brains in a single body, right? Let's take a look at the second brain first. It has glial cells, 500 million neurons, 40 neurotransmitters that are identified, and produces 50% of all dopamine and 95% of all serotonin. Now remember, these are not for the brain in the central nervous system. This is for its own use as a brain in itself. Right? So it's time for you to forget the fact that, oh, are these going to be used by the brain? No, this is going to be used by your second brain, right? It is a brain in itself. So these are its own neurotransmitters. These are what it produces for itself in order to function. That's why I said it requires really a shift in perspective to treat it as a brain, right? The other brain actually has about 86 billion neurons, and for those of you who are curious, really 65 billion of those are contained in your cerebellum, so you have really only very few that are in your neocortex. Now, the finding, of course, this was in, I think, 2012, is that there's a lymphatic system in the brain. We used to think that there was no lymphatic system in the brain at all, and it turns out that the glial cells actually form a perilymphatic space or a perivascular space around the blood vessels and actually cleans out your brain, especially during sleep. So this is a Nobel-worthy development that they actually saw that the brain has actually a lymphatic system. Now, the structure of the brain, all of us talk about the blood-brain barrier as if it were the only barrier of the brain. It is not, right? There are barriers at the surface of the brain, the arachnoid barrier. There's a blood CSF barrier closer to the cavities of your brain called the ventricles. And of course, there's the blood-brain barrier. So all of these are actually significant in what we're discussing today. So when someone says blood-brain barrier, do you mean just the blood-brain barrier, or you mean the blood cerebrospinal fluid barrier, or the barrier at the surface of the brain? These are very important distinctions to make. Now, the function of the brain is for cognition and emotional regulation, and it's actually interesting. Psychedelic research find the ego exists in the default mode network. This is a network that looks like a butterfly like this. It's composed of the posterior cingulate gyrus, the prefrontal cortex, angular gyri. And you could see here that you should keep in mind we're talking about gut microbiota here, and the thing that you could think about is your ego is actually getting controlled by your gut microbiota sometimes. The other thing is that the brain circuit for spirituality has been identified, and it's the periaqueductal gray. And for those of you who administer anesthesia at one point or the other, this is an opiate-rich center that gets you high. And so there is your brain circuit for spirituality. And it's also in the brain, and so this is also being affected by your microbiota. I wonder if anyone here would sequence the gut microbiota of highly spiritual people and see what they get in terms of organisms. Now, the new perspective that I'd like you to go home with is that the brain is not immune privileged. It has a neuroimmune system. You guys are actually very familiar with this. This is the capture, rolling, activation, adhesion. This is all white blood cell activity, right? And it actually can insinuate itself into the neurons via the perivascular space. You could see here, there's a neuroimmune crosstalk, right? There's a healthy brain and a diseased brain. You could see in the diseased brain or the unhealthy brain, right? There's a reactive astrocyte and activated microglia. Remember that astrocytes are just what? Special types of microglia. So let's take a look at the bidirectional communication in the tripartite axis, right? So there's neural, endocrine, immune, metabolite signaling, and vesicular, right? Here you have the neural mechanism. Here's the neural, mainly mediated as is often referenced as a vagus nerve, right? And then the endocrine is via the HPA axis with the cortisol affecting gut motility, the immune system with the cytokines, metabolite signaling, and one more. Here's the bidirectional communication. We all know about the vagus nerve. Vagus actually slows down intestinal motility, and that's a signal. But what we should not forget is they're communicating with the enteric neurons. That's important. We all think it's just coming from the brain. Your second brain is actually communicating with your first brain in terms of its own enteric neurons, okay? Now, there's a bidirectional communication via the endocrine system. You have the hypothalamo-pituitary axis, of course, the cortisol release, and so on. And those actually basically affect your microbiota, and what your microbiota do actually produce metabolites that then are sent back to the brain. If you take a look here, you have we always think, again, this is what I'm gonna emphasize here, you always think that it's coming from the brain. But the gut itself has its own enteroendocrine cells. It means it produces its own hormones like GLP-1, PYY, CCK. These are all enteroendocrine hormones that are produced by the enteroendocrine cells in the gut. As I said earlier, this is a separate brain in itself. So it has its own neurohormones, it has its own hormones that affect it and that affect the first brain in itself. The third is bidirectional communication using the immune system. Of course, here the immune cells, right? They produce the inflammatory cytokines and they affect the brain. If they are inflammatory cytokines, of course, then you will begin to have something like neuroinflammation, right? It says here that the LPS activates an inflammatory response. You all are familiar with LPS. And one of the things I'd like to say here is that cardiologists like to basically bring down your low-density lipoproteins, but your low-density lipoproteins are actually responsible for mopping up the LPS in your system. So there is a certain amount. If you push it way too low, your patient will actually be in a chronic state of inflammation because what? The LPS could not be cleared out properly. Now, metabolite signaling, you know that the bacteria, for example, produces short-chain fatty acids, right? And the short-chain fatty acids, as you will see in later slides, actually is responsible for the behavior of your blood-brain barrier. And among your short-chain fatty acids, the most prominent one that you know about is butyrate, right? And there you have seen studies where low butyrate actually is very much associated with colon cancer. When I lecture to my students, I tell them that if you're looking for a colonic surgeon, you should ask your colon surgeon what the primary fuel is of colonocytes. If he says glucose, you should ditch him and find another one. These are very simple tests. Ask what's the primary fuel, your GI, what the primary fuel of your enterocytes are. If he says glucose, ditch him. Or ask your cardiologist what the primary fuel of the heart cells are. And if he says glucose, ditch him. If you ask your neurologist what's the primary fuel of the brain at rest, if he says glucose, fire him. And these are basic or fundamental things that we all should know, right? Both doctors and practitioners should be very aware of what fuels your fundamental cells. So it's the neural, it's the vagus and the enteric nervous system. Endocrine is the hypothalamo-pituitary axis and the enteroendocrine system. Then there's immune signaling, metabolite signaling, right? And then the last one actually is by what's called bacterial membrane vesicles or outer membrane vesicles. So that again, the communication is bidirectional, that the bacteria actually produce membrane vesicles that can then fuse with your enterocytes. And it may contain anything. It may contain RNA. In fact, the Nobel Award for this was showing it containing RNA. So for those of you who get questions about COVID vaccine, right, that it actually has RNA and it's being incorporated in the system, evolution has been doing this for us thousands of years in our gut. So that's just something for you guys to think about. Now, here's what I was telling you about the bidirectional communication in the EMGB axis, right? This was in 2013. And he says, recently inter-kingdom crosstalk was demonstrated between humans and bacteria through fecal microRNAs. Why is that significant? Because interspecies talk is not allowed, right? It's just like reproducing between a hippopotamus and a swallow, for example. But now we find that they actually communicate, right? And we talk back and forth with them through RNA. They alter our epigenome, and guess what? And therefore, they will alter our behavior as well. So the way to look at it is what I term as a holobiont. We are basically an assemblage of organisms. We are a network of organisms that are all communicating with each other. We cross a species in order to talk to each other, right? So human cells, human microbiota, food, fungi, and other viruses, they are all in a network within our body as an ecosystem, and they all talk to each other by different means. And the last means that was just shown was actually by membrane vesicles, which was forbidden. This form of communication between species was forbidden by biology, by dogma a long time ago. Finally, this is a 2019 article. They're finally using the term holobiont in the Microbiota-Gut-Brain axis articles that you will find out there. Now, the third part would be an introduction to home hope for those of you who weren't here yesterday, and then how to detect and correct imbalances in the enteric microbiota-gut-brain axis, and then enteric microbiota-brain interactions, the interaction between the two organs, right? And then neuroinflammation and illness medicine versus health optimization medicine and practice. This is a very simple way to display what we do in health optimization medicine. In illness medicine, essentially, you look for disease markers like FBS or HbA1c, right? And then you basically correlate it with some disease and you do disease management. So it's very simple. Here we do diagnosis and treatment, and that's the clinical practice of illness medicine, is to diagnose and treat diseases. However, with the advent of technology, we now know how to look inside the cell, and by looking at it, we see that actually there are about 4 drivers that precede the disease markers, right? These are genes, symbiotic partners, environment, and time. For genes, you have the various types of the nuclear DNA, mitochondrial DNA, nucleic acids, your symbiotic partners, your mitochondria, which are endosymbionts, bacteria that live inside your cells, your gut microbiota, which are your exosymbionts, bacteria that live outside of your cells, your environment, like the direct and indirect signals from environment. For example, light can be phototoxic to you. And then evolutionary and circadian time. And all of these gears actually produce metabolites that can be then detected and corrected, right? And you can detect and correct metabolite imbalances, and that's what we do at Health Optimization Medicine as a clinical practice, is to detect and correct imbalances. We really don't do disease. We set the diseases aside and make sure we take care of your fundamental cell first in terms of its balance. If you want to check for the health markers and adjust the metabolome itself, then use the science of clinical metabolomics. If you want to affect the DNA, we're too old for CRISPR-Cas9 technology. Trust me, I tried. I bought a CRISPR-Cas9 lab for myself and was unsuccessful. All I was able to do was develop a glowing Saccharomyces boulardii. But anyway, if you want to affect the DNA, then there's the science of epigenetics. Then if you want to affect the mitochondria and gut microbiota, you use the sciences of bioenergetics and the gut immune system. And if you want to affect the direct and indirect signals from environment, use a science called the exposome. Exposome, which is the sum total of your exposure to everything, right? And my favorite example here is that if a mother drinks during a pregnancy, then that's exposure to alcohol, right? And now you know why we look at this history when we're trying to figure out whether or not we have a fetal alcohol syndrome or an adult that was exhibited fetal alcohol syndrome as a child. If you are looking to change the evolutionary and circadian time, you use the sciences of evolutionary medicine and chronobiology. Take a look at the ohms here, right? Metabolome, mitochondrial metabolome, gut microbiome, epigenome, cell danger response metabolome for evolutionary medicine, and in chronobiology, we have the chronobiome, and then you have the exposome. So these are the 7 pillars of health optimization, medicine, and practice. Unfortunately, this is not taught to us in medical schools yet, but we hope that by doing this and educating a lot more people, we can get to where we want to be in terms of health in this country and in the world. Let's define health. If you ask any person, especially a doctor, to define health, they wouldn't have any definition of health. I like a more clinically actionable definition of health, and this is, you can take this with you. A plus B plus C is the absence of disease, which is illness medicine, and the balance between anabolism and catabolism according to the cycle of life of the organism. And we don't do root causes of disease. There's only one root cause of health, and that's the maintenance of balance between anabolism and catabolism, right? And I hope someone now here knows the answer to my question yesterday. Is inflammation anabolic or catabolic? I'm not going to answer that for you. So it means that just because you're not sick doesn't mean you're healthy. It only means that you're not sick, right? And optimization now is defined as neotenizing your metabolic subnetworks to values that it had when you were between 21 to 30 years old, or use evolutionarily derived values, right? And neotenizing means making young. When your patient or client comes to you and says, oh, but this value is normal, that's normal for the age. But what we try to do, or what we do, is actually to move your values to the values you had between 21 and 30, the golden age for have children. Then Hertog actually did this in hormone balancing. He uses a mean value of 25 plus minus 2 standard deviations. United States, by the way, uses 30 for hormone. Right? Gordon, his median value, he takes a median value between age 20 to 30, and I use the 50th to 75th percentile at age 21 to 30, and I use evolutionarily derived values, for example, for vitamin D. Vitamin D values, as I see in this country, the reference range is way too low. We do not use illness medicine range of values. Lab values are cutoff values. They are survival values and are not optimal values, and normal is not optimal. Now, the practice is very simple. You measure the levels of metabolites, you compare them to the optimal ranges, not the normal ranges, and you balance using 2 major techniques, which is network-wide range shifting and ratio correction. Network-wide range shifting brings the concept of network. Everything is networked in the body. If you touch one node, everything else moves. The other one is ratio correction. For example, if you are looking at omega-3, omega-6 balance, then you should correct this by correcting the ratio and not correcting a single value in those metabolites. So this is a visual summary of home-hope balancing. We all know that oxidative stress leads to all of these diseases. It's not only oxidative stress, right? There's also the nitrogen stress, the RNS, right? Which is used by the body really to kill viruses instantly. And so here it is, there's a redox balance and redox signaling. So we're after balancing. See here the mitochondrial homeostasis, metabolism, inflammation on the other side. And then if you take a look, this is the yin-yang of inflammation, right? For if you have imbalanced redox, then your NF-kappa B transcription factor gets activated and immediately produces your pro-inflammatory cytokines, right? There's NF-kappa B activation. Your body knows what to do. Yeah, we're not dumb organisms. We evolved over time. We have our own mechanisms of healing and protecting ourselves. We should take a look at what we're doing first before we actually go gung-ho and do different things to the body by shoving all sorts of things to it. We should know what the body is doing evolutionarily before you start changing things. The NRF2 system produces anti-inflammatory cytokines, and this is the yin-yang balance of inflammation and anti-inflammation. Many of you like turmeric. It's a very common way to decrease total body inflammation because it is a direct activator of the NRF2 system. Now, here's your pro-inflammatory, anti-inflammatory balance. You can do a cytokine testing if you like to, or if your patient is rich enough to have this done. Again, you take a look at the balance of your inflammatory and anti-inflammatory cytokines. In homobalancing, this is like the center of what we do. We optimize the resolution of the cell danger response, right? There is cell danger response consists of 4 major parts: oxidative stress, which you could see can be the trigger. It's like your reactive oxygen species or reactive nitrogen species, right? And then your amplifier would be the inflammation via your cytokines. And then there's innate immunity, the activation of, say, your macrophages, right? And then there's the endoplasmic reticular stress response. As I said yesterday, you guys must be very familiar with this. This is unfolded protein response. This was elucidated over 2 decades ago, and you guys should be experts on this by now. This acute physical, biological, and psychological threat induces the cell danger response, right? And so does chronic physical, chemical, and biological threat. Acute threats, usually we can resolve it by ourselves, but the chronic threats are the ones that produce chronic diseases like diabetes or clinical cognitive decline. So I asked ChatGPT-4, said, are there any vitamins, minerals, cofactors, and hormones that dampen or quell the cell danger response? And it says vitamin D, C, E, A, minerals, zinc, magnesium, selenium, cofactors, CoQ10, ALA, NAD+, hormones, melatonin, cortisol, and testosterone, and others, omega-3 fats, and polyphenols, right? But should you give everything if your patient is, or client is in a cell danger response mode? Of course not. What we do in health optimization medicine is actually we measure, right? So we measure the levels of antioxidants, vitamins C, A, alpha-lipoic acid. Those are measurable right now by clinical metabolomic testing, right? So before giving any of that stuff, just because you have ChatGPT-4 doesn't mean you should just inundate your patient with all the supplements. Measure and see whether or not they need them, right? If your patient is in actual anti-oxidative stress, then they are warranted to the levels your tests are showing. Remember that there is a syndrome called AIS, antioxidant-induced stress. If you give way too many antioxidants, your system will go haywire. It will go the other way, right? And why is this? For example, if you enlarge the pool of your antioxidant vitamin E, that's a very large pool. And remember, they're in a cycle. Whatever is next in the cycle is still a small pool. So when you're looking at increasing your antioxidant pool, for example, take a look at your antioxidant regeneration pathway, right? Because all of those are involved. What you're going to do is simply pool the vitamin E and what it has reduced in its pool, and then a smaller pool is waiting to actually do the recycling for it. And that's the reason why many studies on, for example, selenium and vitamin E failed in 80,000 cohort for prostate cancer. If you don't measure this stuff before giving them it, you are doing a wrong protocol, right? And then the imbalance of enteric microbiota is called dysbiosis, of course, you know this, and you can detect and correct, for example, malabsorption and bacterial dysbiosis types of metabolites, right? You know how to do that, and you could do your recommended supplementation and dosing with prebiotics, probiotics, parabiotics, postbiotics, symbiotics, high fiber diets. As I say in my lectures, the fastest diminishing macronutrient in the human diet is fiber, right? Lifestyle adjustments like meal timing. All of us usually sacrifice our meal timing in order to do our work or to have our extracurricular activities. So let's take a look at the enteric microbiota and the blood-brain barrier. You can see here the role of short-chain fatty acids. As you can see, short-chain fatty acids, you have to have the levels such that they actually promote strengthen the blood-brain barrier. It has an anti-inflammatory effect. It's neuroprotective. It regulates the microglia. Some of the SFCAs can actually cross the blood-brain barrier, right? And it's an area of ongoing research, and it can serve as an energy source in the brain, such as is the primary energy source for your colonocytes. When you're supplementing, for example, the short-chain fatty acids directly and not via high-fiber diet modification or probiotic modification, then you should make sure that the short-chain fatty acid actually is enteric-coated enough to actually get uncoated or dissolved in your colon, right? Otherwise, it's basically useless. Now, the enteric microbiota and brain neurotransmitters, as you can see here, essentially the neurotransmitters, SCFAs and BDNF. So the neurotransmitters, serotonin, 90% of serotonin is produced in the gut, but remember, serotonin is not produced, does not cross the blood-brain barrier. So the serotonin in the brain is produced by the brain itself. You have your tryptophan. There's a large neutral amino acid transporter that actually transports tryptophan and creates its own serotonin. So this serotonin is actually produced for the use of the gut itself, right? And then dopamine, some bacteria can produce dopamine and they can have their systemic effects. We don't know whether or not it crosses the blood-brain barrier. I suspect it doesn't. GABA, of course, is notoriously a huge molecule. It does not cross the blood-brain barrier in a classic sense, and therefore it has to be synthesized from scratch via the glutamine from your diet, which becomes glutamate in the brain and then gets converted into GABA. Now, certain strains of bacilli and bifidobacteria can actually produce GABA. And then norepinephrine is also produced by gut bacteria. So again, the main perspective here, even if you don't remember any of this, is that the gut is itself a second brain, right? It has its own neurotransmitters, its own hormones, and you should respect it as that. So the enteric microbiota effects on BDNF, you have BDNF upregulation, stress response. If you have stress-induced changes in your gut, it will actually decrease your BDNF. And for inflammation, of course, gut microbiota can modulate systemic inflammation, which can in turn influence BDNF levels. What if your patient or client complains that he or she is so poor, could not buy all of the prebiotics and probiotics that you recommend? One of the biggest things that you could do is actually exercise. It can raise your BDNF by about 60% and beats the results of any SSRI that you could give out there for depression. That is, if you could push them to exercise. Your microbe-associated molecular patterns, right? And then you have DAMPs, damage-associated molecular patterns. You guys are familiar with those, right? Those are old terminology, PAMPs and DAMPs. And you could see here that microbiota actually modulate your neuroimmune function. And implications of the modulation of neuroimmune function, actually altered gut microbiota can lead to increased levels of pro-inflammatory cytokines, right? When you look at immunity, when a patient presents to you or a client presents to you, for example, with lupus or some autoimmune system, don't look immediately for autoimmune causes. Look right away at the gut health. What can you do immediately for your patient or client is actually to clean up the gut, seal it up, and make sure that the microbiota are appropriately replenished. Autoimmunity, neurodegeneration, they're implicated in neuroinflammatory aspects of Alzheimer's and Parkinson's. I have a few of these patients, and one of the first things that I do really is to clean up the gut and decrease total body inflammation, because an inflammation in the gut will already give you a clue that the brain is also inflamed. Question again, how does one take care of gut dysbiosis? Dietary changes like prebiotic foods, probiotic foods, low sugar and low fat, supplements, probiotics, prebiotics, digestive enzymes, glutamine, berberine, caprylic acid, zinc, carnosine, curcumin, lifestyle changes, stress management, physical exercise, adequate sleep, medical treatments, right? If antibiotics are warranted, I prefer to use natural antibiotics, and antifungals, right? And fecal microbiota transplants. Those have been in the news over 5 years ago. These are classes of what actions you can think about for your patient or client: dietary changes, supplements, lifestyle changes, and if warranted, any medical treatments. So, this is a simple diagram. You already know what this means. Probiotics are healthful microbial living cells. Parabiotics are dead or inactivated probiotics. And then prebiotics are chemical agents that have the ability to stimulate probiotics. Postbiotics are beneficial metabolites of probiotics. Prebiotics and modulation of the microbiota-gut-brain axis. So that's how, this is from this paper, and that's how I'd like you to remember the gut. It's not as a gut, but microbiota-gut-brain axis, gut-associated lymphoid tissue, because that's where most of the problems of your patients or clients will be coming from. Think of it as really an immune system. And as I say to my guys in health optimization medicine, your immune system is responsible for your defense. And if you look at the United States, the Department of Defense has the biggest budget. Energy budget of the body is also the biggest for its immune system. All right. So again, there's the imbalance of the gut immune system. We have a leaky gut. You guys are very familiar with this. As you can see here, there are factors that can cause decreased permeability, like propionate, butyrate, oligosaccharides, fiber, increase with phenylenedic acid, propionate, glucose, fructose. So always have that balancing principle in mind when you're looking at the gut or the brain or any other part of the body. And if there is too little permeability, then you cannot absorb anything, right? It's too much, then you have what's called the leaky gut. So what you have in here is you achieve a balance between an optimal type of permeability for your client. And this is very individual because the lifestyle, the diet of your clients usually stay in one place. And then it's usually very steady. If it's continuously very fatty, and you guys know that the most difficult thing to change in any client or patient is behavior, right? So you have to follow their behavior for a while adjusting the permeability of their gut. It's not a one-size-fits-all thing when you're looking at the client, right? So and then you could measure calprotectin, EPX, fecal secretory IgA, the butyrate, propionate, acetate. Take a look at those levels and determine appropriate supplementation. Now you could do your elimination diets, your glutamine, probiotics, digestive enzymes, etc. One of the things that induces leaky gut actually from stress is post-operative patients. When you're dealing with a post-operative case and they usually just dump the patients in the recovery room and give them nothing by way of any support. And when you receive the patient is in severe leaky gut and complaining of other problems. So what you can do there is to recommend 6 grams of glutamine in a liter of water to be sipped by the patient while in the OR, especially if intestinal surgery, right? And that actually keeps the patient in positive nitrogen balance, right? And that will make sure that your intestinal permeability is not harmed that much. It will still, any surgery to the body is still stressful, right? How does one take care of the leaky gut syndrome? So you don't need me for this anymore, you just need ChatGPT-4. Dietary changes like the elimination diet, high fiber foods, probiotic-rich foods, supplements, probiotics, glutamine, digestive enzymes, omega-3s, zinc, collagen or bone broth, right? Vitamin D, turmeric, lifestyle changes, stress management, if they need to divorce their husbands or wives, right? Adequate sleep, right? If the partner is snoring, it's been shown that for each snore, the cortisol level rises in your partner. Each snore that your partner makes, your cortisol actually rises. You may be in love, but you're sleep divorced. And then regular exercise, which is one of the most difficult things to get your clients to do unless they're actually younger and seeking a mate. Now, medical treatments, as I said, only if needed, antibiotics, antifungals, anti-inflammatories, right? And what I want you to note here is simply the classes: dietary changes, supplements, lifestyle changes, medical treatments. We're looking at brain balance when you go take a look at neuroinflammation, right? It's a balance of microglia. They're either activated or inactivated. A resting microglia will have a regular phagocytic function. They will have steady state monitoring. Remember, this is the immune system. They're soldiers. They're sentinels. They monitor their environment, even if you consider them as brain cells. They serve as the immune function. And then there's the activated microglia, which now produces your pro-inflammatory cytokines. And you could see here, there's an dysfunction, neural injury. I went quickly through a slide earlier that showed the cell danger response. Now, what I wanted to emphasize there is the following. There are only 8 ways by which a cell, a fundamental cell, can respond to stress, evolutionarily. All the other ways that it responds are actually due to their specialized functions. And that's what we actually teach in our online module on evolutionary medicine, is the cell danger response metabolome, right? There are only 8 of those. And if you could actually resolve those 8 different steps, you actually have already resolved the cell danger response that the stress is putting out, and you have quelled it, and your patient or client would start feeling better, right? Now, here's a cell with no neuroinflammation. There is an oval for inflammasome. Unless you've been living under a rock, you all know what an inflammasome is. The most famous one, of course, is the NLRP3 inflammasome, right? This is the staging area for your inflammation. As you can see, everything is nice and neat and clean. And when you look at the gut, you should always think about the brain, right? Because they have the same fundamental structures. You have your bloodstream, you have your endothelium, and you could see that the lymphoid tissues are underneath the gut, whereas in the brain, your glial system actually serves as the immune system. So always think of these two in parallel, especially if your patients or clients are exhibiting brain fog at the same time, right? You might think, oh, that's thyroid. No, it may just be a simple neuroinflammation from a gut dysbiosis, right? Don't jump into conclusions immediately that your patients need any of these big guns right away. Now, see here, take a look at neuroinflammation. They will occur almost at the same time if you don't address the inflammation of the gut. Inflammation of the gut usually occurs first because that is what is exposed to food, right? External elements. So the body has to fight what it doesn't want inside and actually allow what it wants to get inside. And you could see here is a total inflammation. And when I say inflammation, not the traditional mosquito bite inflammation should come to your mind. It should be what's called molecular inflammation. What you should think about is that there's heavy reactive oxygen species load, your NF-kappa B is activated, your cytokines are being produced. That's the kind of inflammation that should be in your head. We're no longer in the 1940s where you have to look at microscopes to do that, right? We deal with metabolites now. And you could see in the brain itself, you could see that your glial cells are the ones responding as your lymphoid tissue in the brain, and they alter your neurotransmitters, right? ChatGPT-4, like what can be done to dampen or quell inflammasome activity? It says here, natural compounds like curcumin and resveratrol, lifestyle and diet like fasting and caloric restriction, an anti-inflammatory diet, microbiota modulation, probiotics and prebiotics, fecal microbiota transplant. I've had cases where I actually had to send or accompany clients in Germany for adult fecal microbiota transplant for C. diff, severe C. diff in adulthood. And epigenetic modulation metabolomic approaches, which is what we actually do in home. So not all neuroinflammation is bad. Your gut, there's a certain optimal form of leakiness. In the gut, there's also an optimal form of inflammation that's optimal to the brain. So on low inflammation, there is immune to brain communication with immune surveillance. There is involved in development, memory and learning. Transient inflammation is involved with injury and induced remodeling. And this is called eustress. This is inflammation, right? And for those who are stoics here, eudaimonia. Anyway, negative aspects of neuroinflammation, as you could see, these are high inflammatory states. And again, when you think of inflammation, think molecular, right? Your mitochondria are highly stressed out. They will put out a lot of reactive species. They will induce your cytokine production. Those will activate your NF-kappa B. They will induce your cytokine production. And then you will have all the downstream effects of molecular inflammation. All right. Now, what's wrong with this picture? Can anyone tell me what's wrong with this picture? Microbiota and the gut-brain axis, implications for a new therapeutic design in CNS. This was just published in 2022. So this is beautiful, right? Microbial metabolite entry, altered receptor activity, altered neurotransmission. The pathways is what we discussed: immune, neuroendocrine, enteric nervous system, circulatory system, vagus, secretions, peptides, gut hormones. Very nicely done, right? And then up there, you have your neurodegenerative diseases like MS, Parkinson's, Alzheimer's, brain tumors, and then neurodevelopmental and neuropsychiatric disorders like autism, and depression, and anxiety, and stress, and schizophrenia. They immediately proceed to disease, right? Many of your clients or patients will not be having these things beforehand, right? These things come as a consequence of what happens beforehand. So if you could catch that for them beforehand or prevent that beforehand, why would you not do it if you already have the tools and the products to do it, right? So it's not always about disease. It's time to think of health first. So when you see a client, you set aside what their diagnoses are and take a look at what the fundamental or basic cell is doing. Is the mitochondria all right? Is the cytoplasm okay? Are the cell membranes okay? Is there very fast oxidation of the cell membranes? Is there any indication that the DNA, nuclear DNA, is oxidizing very rapidly? You could see all of those in a fundamental cell, and all cells of the body have that, except of course for mature red blood cells which don't have mitochondria. So you could see here that diet, genetics, and epigenetics, mode of delivery, environment, drugs, and exercise affect the enteric microbiota-gut-brain axis, and it will affect the behavior, food intake, cognitive behavior, stress, social interaction, fear. And as I said earlier, it can affect your enlightenment because it affects your default mode network. It can affect your spirituality because it affects your periaqueductal gray. Take care of this organ that grows after you're born or after your patients or clients are born. Let me just show you the difference in care between illness medicine and health optimization medicine on the Enteric Microbiota-Gut-Brain axis, right? The first one is a diagnosed disease. So in illness medicine, you have anxiety, depression, multiple sclerosis, Parkinson's, Alzheimer's. Whereas what we do is we just detect imbalances. We use metabolomic tests to detect imbalances in vitamin, mineral, cofactors, carbohydrate, protein, fat, hormone metabolism, detect exposure to toxins, etc. And then you use the enteric microbiota. Your goal there is to achieve eubiosis. For us, we want to correct imbalances with supplementation, diet, exercise, sleep, and other lifestyle advice, right? For illness medicine, you want to seal the leaky gut. And for us, we want to resolve the enteric neuroinflammation. Inflammation is not only in the brain, guys. Remember that you have a second brain, and therefore you have an enteric neuroinflammation. All right? Don't forget these things. When you remember, this become very fundamental to your care because you will remember immediately what to do with your patients or clients, right? Oh, I'm not only dealing with neuroinflammation here, I'm also dealing with brain inflammation, right? Because they will tend to co-occur after a while. In the brain, you treat the disease, like with drugs, right? For Parkinson's, for example. And in the brain, you resolve the CNS neuroinflammation, which includes achieving eubiosis and sealing the gut. Now, for those of you who have Parkinson's patients or are Parkinson's clients, you already know that the Parkinson's is now considered to have the dual physiologic pathways. One is actually via the gut, and the other one is central. You could see now that we are seeing the gut-brain connection even in the diseases themselves. Home hope, take care of the needs of the fundamental cell and its network first, and the rest will follow, because disease is managed, but health is optimized. Managing means that you're only there until the disease resolves, but health, you're there every day. And my favorite question here from an audience one time is asking me whether or not we could bank exercise. Actually, you can't, and that's just like health optimization, right? So the order of use of balancing agents, the first line will be your bioidenticals and bioactives, like bioidentical hormones, bioactive nutrients. An example here would be using methylcobalamin instead of cyanocobalamin. Cobalamin is an artifact of manufacture. Cyanide is an artifact of manufacture in cyanocobalamin. Second line would be your phytoceuticals, fungiceuticals, bacteria, like your probiotics, phages. And the third line would be xenobiotics. And why do I put drugs last, even if I'm trained in pharmacology? Is because almost all the drugs that we have out there have never been encountered by the body in evolution, and the body doesn't know what to do with it. But it knows what to do with your hormones, nutrients, and other things that it actually uses to heal itself or to balance itself. So use those first, and use the big guns later, because you know when you start to use a synthetic antibiotic, you're going to bomb your gut microbiota organ. So this is the balancing, very simple. You measure the levels of your vitamins, minerals, cofactors, toxins, and neurotransmitters, and so on, and then you give them measured supplements. So the era of asking me whether or not vitamin E is good for you is over, right? You can measure all of those things, and they can be measured at a reasonable price. Now, the latest developments we have in these particular areas, the dude herself is now in the game. She's a Nobel laureate in the CRISPR-Cas9 technology, right? And watch out, guys, she's ready to tackle gut microbiota and changing the genome according to what you want them to produce. Whether or not, are we going to have non-GMO microbiota? I have no idea, but she's already expressed her interest in the game. They found that there's a dedicated cytoplasmic container that collects exosomal DNA away from the mammalian nucleus. And it's a new organelle. It's called the exclusome. So you now know that we have another organelle in the cell, and it's called the exclusome, right? And the latest developments in the brain, a new kind of cell shakes up neuroscience. And we continually find newer and newer cells, right? The specialized astrocytes mediate glutamatergic glial transmission in the CNS. So the new organelle is called the exosome. This is called the what? This is called the glutamatergic astrocytes. Astrocytes are not supposed to produce any neurohormones, but this one produces actually glutamate, all right? So the latest developments in the Enteric Microbiota-Gut-Brain axis, Kieran yesterday was discussing how you develop drugs and how you do test various types of microbiota in the SHIME system. Now they're going ahead rather than using the SHIME system. What I want you to see here is that one. And the term that I'd like you to remember is organoids. They're now using intestinal organoids and brain organoids to test what particular microbiota are producing what particular metabolites that will affect the brain, okay? And this is big news. This is a totally different way of doing things now. We've been doing things forever via the SHIME system. So the latest developments in AI, I created this chatbot for us who actually do not like to memorize things and just like to seek things out. This is what's coming. We intend to give this to our practitioners, health optimization medicine and health optimization practice practitioners. Initially, right, this says what to do if uric acid is high. And I threw in a few books in there, and KB stands for knowledge base. And this is what it came up with after looking at the books that I put in there. If uric acid is high, it may be indicative of abnormalities in arginine metabolism. And then I actually placed in a separate window the ChatGPT response itself. As you can see, if your books are in here, the professional books, the response is a lot more professional to your liking. ChatGPT is slightly a little bit more general, but can provide you insights as to the other things that you may want to investigate. So this tool will be available hopefully next year to our practitioners. And a lot of AI is actually already being used now for the gut microbiota and the development of newer probiotics, okay? Here's our summary. This is my ending slide here. All the things that we talked about, the enteric microbiota, gut-brain axis, the ways by which they communicate, what can go wrong with the communication is usually mediation by inflammation, right? And so the ability of brain to influence the intestinal microbiota, that's one way down, and the ability of microbiota is to go the other way up in a bidirectional way. Don't forget that the classic things that you learned about have their counterparts in the gut, like the vagus has the enteric nervous system, right? The HPA axis has your enteroendocrine system, right? So think of it really as another brain that actually has its own functions. It's what gives you your butterflies in your stomach, right, when you're nervous or when you're about to divorce your spouse. So when Home Hope takes care of the needs of the fundamental cell and its networks first, and the rest will follow. Remember that disease is managed, health is optimized, and encourage you to optimize the health of your patients or clients rather than managing their disease. Thank you very much. This is my last slide.